So hello, my name is Joe Muldoon. Thanks so much to the GMOS organizers for the opportunity to speak today about a topic that I think a lot of us may have heard not too long ago. Actually, I guess it was, but it feels recent. So in the summer of 2015, we had a really great talk by Professor Lori Zoll. At the time when this thing called the Human Genome Project, right, or HGP, right, was just coming online, there was a really high impact meeting was held. We had this super cool conversation about all the ethical and philosophical implications of it. So it had been a while. I thought, it's been almost 2 years. What's going on in this space? Let's maybe reopen this conversation and see what's happened since. So first thing to note is that it's no longer HGP, it's GP. So this is Genome Project Right, which implies like underlying fundamental change in a lot of the organization or the shift here. So what I'm hopefully going to show you today is where this project is now. It's certainly still getting up and running, but hopefully we can get a glimpse into what's coming up. As we go, as always, this is GMODs. It's informal. Please jump in, interrupt, raise your hands. I'm going to present very factually what's the case, not my own opinions. So I welcome your own thoughts as we go. Sound good? All right. Okay, so before we talk about right, let's just recall where we came from in our history here. So the Human Genome Project READ, as it's kind of retrospectively being labeled right now, it was initiated a few decades ago. In 1990, the NIH, there was the genome basically section of it led by Francis Collins, started this public sector effort in order to sequence a human genome. And the approach they used was this thing called hierarchical shotgun sequence, where basically you say the genome is pretty intimidatingly large, let's break it up into these smaller chunks, and then we'll deal with one chunk at a time, and then at the very end, we'll assemble them bioinformatically. So it wasn't until quite a few years later that Craig Venter left Tiger, the Institute for Genomic Research, and started a new company that later became Celera. And this was basically, at least as was presented in the news, the competing private effort. And they used a slightly different approach called whole genome shotgun, where you basically say, nah, it'll be fine. Let's just sequence the whole genome and we'll do it all at once. So when you consider when these two started and when they finished, there was basically an effective tie that was announced or co-announced in 2000, where they both said, let's just call it a tie. We basically finished the genome. But what does it really mean to finish a genome? Because 3 years later, they say, oh, well, this is the more complete version. And this is kind of a never-ending story because you have the A's, C's, G's, and T's, but what does it all mean? We get things like the ENCODE project, where we try to decipher, not just in humans, but also in other organisms, all the functional elements in the genome, so enhancers, promoters, and so forth. And shortly after that, there was this 1KGP, 1,000 genomes project, where they said, okay, well, guess whose genomes were sequenced originally? If we were charged with right to sequence any genome, you'd probably sequence your own, which is what went down. So to try to get more diversity of humans worldwide, they pulled people from all different continents to do that. So this is the background for reading lots of genomes. And so it wasn't until 2015 that an international community started to coalesce and said, let's talk about writing genomes. And not just writing any one genome, but synthesizing, writing, and then testing in living cells over 1,000 genomes within the next decade across cell lines and finding some way to dramatically reduce the costs. So this is a pretty cool, ambitious goal. Although now it's called GP-Write, the human genome is still a core component within that umbrella, as well as a lot of other genomes, like plants and agriculture relevance. Just to clarify, there's no designer humans being created here. All this work is in a dish. So cells, tissues, and organs. So that is stressed very strongly on every corner of the webpage. Yeah. All right, and then quite humbly, the group that's carrying this out is the Center of Excellence for Engineering Biology. This is their logo. And then but participants can come from any sector. So at the end of the talk today, we'll talk about some of the proposals that have been submitted. And I'm pretty sure anyone in this room with their PI can submit a proposal as well. It's quite straightforward, 2 pages, similar to 1 page, 3 paragraphs. We propose to build the genome. You get the check mark in your money. So it looks promising. Okay, so far the people who have expressed interest in becoming involved, this is a very kind of do-it-yourself apply as you would in any grant situation, have come from many different places. It's not just the US. I think there are at least 15 countries here, probably more. It's not a little outdated. And as of 2017, they can count the following numbers. So a couple $100 million it's been made available to different institutions, I guess, for people to start their own projects. The number of attendees at these meetings seem to be capped by how many can fit in the room. I suspect many more people wanted to attend. A lot of working group committee members. So among those are Josh Leonard and Danielle Palmer-Grisek here. Those are the two NU professors who are involved at a high level with this. And yeah, I suppose there'll be lots of videos online. Other facts, cheesy countdown to one international community taking on the Grand Challenge. But yeah, so very, very optimistic vibe. Okay, so we often see polls about the public. And so I thought this was very interesting in that this was a poll for attendees based upon the 2017 meeting. So in these numbers we're going to look at here, we're basically getting a cross-section of what other scientists think. So I really appreciate this because it's like, essentially, if you have the most possible information, how would we expect people to think about these issues, as opposed to the larger cross-section? So what is the single greatest hurdle? A lot of options here, but in bold, you can see that a lot of people are focused on the cost and funding challenges. There's definitely an issue of public perception, and then technical, I guess, can we actually carry these things out? People aren't concerned about these other issues. Political climate apparently isn't concerning people about the ability to move forward with GPA, which is interesting and good. All right, what's the greatest technical hurdle that impedes our ability to synthesize genomes in ways that we need or want to? DNA delivery. So people are really focused about after we synthesize, after we write, how do we actually get this stuff into a cell? It's one thing to synthesize relatively short regions of DNA, but if you get to anything, I think larger than 30 kb or so, it's really hard to manipulate with that before things work. So people are really focused, I guess, on if you build something, how do you know it won't fall apart? And then one more question. Your greatest concern with respect to synthesizing genomes? Potential misuse and safety. I'm just curious, how do people think this might align overall with the way that the public more broadly might answer these questions? Yeah?
I mean, the public's perception of safety is different than the scientist's perception of safety. It's two different workplaces, I guess. You're not familiar with our workplace.
So their perception of safety, or the public more broadly, how do you think that might differ?
Well, you're talking about safety in the lab and safety in the environment. So they're going to have two different, completely different things.
Yeah, that makes sense.
I'm just amused by like the 9.6%. No concerns. There's no
So of the 250 people at this meeting, a solid 24 not concerned.
Did they all,
I mean, sorry. Oh, go ahead.
did they all answer the poll? Because it seems like there are 3 that have the exact same amount of people. That seems unlikely for a 250 person poll.
Oh, you're right. Probably like, we could back calculate. Good one. So we're getting a slightly biased sample of a highly select group. There's always poll bias. Whoever wants to fill up a poll already feels strong to begin with, except for the last few years. Anyone else? Yeah.
I wonder about the differences between ethical and potential misuse. I think that to the public, so for scientists, I think that the intended use probably has not many ethical qualms, right? The intended use would be solid and good, right? The public might think that even the intended use is the problem. So I think that, I just think that those are sort of, it's interesting that it's broken down, potential misuse is much higher. I guess we're talking about things like bioterrorism and misuse, but I feel like the public would be higher on ethical. I think that split might change depending on who you ask and what they think about the intended use.
Yeah, and if I understand that you're saying, I think if I understood the wording of the question kind of inherently presumes that you're walking with a certain value or value judgment about what ought to be done, right?
Yeah, and about the intended use being the good use.
I also think the public has a drastically different view of the political role that scientists envision themselves playing in the world, if that makes sense. Especially in our current political climate, there's certainly been almost an causing anti-science backlash in certain circles. So I could see some people believing that we are going to be synthesizing genomes for political ends that other scientists would say, I mean, like, what is the point of synthesizing a genome? Like, we're still trying to figure out the pipetting situation.
Making some progress there. Yeah. Cool. Any? Good. Yeah, I think those are all interesting points. Okay. All right. Now the fun part. So on their website, they're being quite transparent about everything. So I definitely invite everyone here to Google GPWrite and look stuff up. They've posted all of the projects so far, the pilot projects that have received funding, and also all of those that have been proposed and are under consideration. So what I want to do is just kind of highlight a couple of these that I thought were particularly interesting and show you what's coming around the corner. So things will maybe be finished within a couple years, but are just getting up and running. As we go, yeah, please continue to jump in. Okay, so there's a list. There are 13 of these. As we go, you'll notice that many of these are not strictly about synthesizing a genome. A lot of them are like fundamental basic science questions we're trying to answer. A lot of these are biting off chunks that are more doable than doing a whole genome, like doing a chromosome or picking one region of a chromosome. So as we go, it's kind of interesting to see that in changing from HGPWrite to GPWrite, it's now further diversifying this whole umbrella of different related projects. Okay, so here's one from Jeffrey Way and Pam Silver. Pam Silver gave a talk here not too long ago. And the proposal here is to synthesize a couple, about 1 million base regions of the human genome in yeast, and then transfer them to humans. That's how you avoid the heading issue. And the long-term goal is to be able to control the expression of genes that are adjacent, driven by different promoters and separated by insulators. So how do you control genes on a very long stretch without something kind of bleeding into what's adjacent to it? Okay, big whoop, right? Synthesize 2 times 10 to the 6th. But it's not just that. They're going to construct a combinatorial library of regions that vary in promoters and insulators for 2 regions, one for the type 1 interferon genes and one for the estrogen responsive genes. And basically do a whole bunch of characterization capturing the 3D chromosomal conformation, gene expression outcomes. And so basically, if you scramble different regions of a whole chunk of DNA, and you try to refactor how they're regulated, how does it behave different from the native thing? So if you're going to take a genome and just totally impose your own control over all of its regulatory regions, does it fall apart or does it withstand that kind of engineering? Okay, here from Jeff Bucca and George Church, they want to make a universal ultra-safe cell line. We're going to make a perfect universal cell line for everything. It's basically how it reads. We're going to do all this stuff. We're going to alter just 1% of the human genome, namely all the exons. These things read really funny. Please do read them. And these are all the properties and the explanations. So it's kind of a wish list of all the things I want to see. I don't know how we're going to get all these, but it looks really cool. So prion resistant, so not going to get neurodegenerative diseases in a dish. Scrambleable, for rapid optimization. Immunonegative, that's really important. So if we're going to synthesize synthetic organs, we don't want to put them in a recipient and then have the whole thing go awry. Cancer resistant. So that sounds, I don't know what you're gonna, as Benji said last time, the only way you're gonna avoid cancer resistance is you make something called unicellular. Okay, so the ideas get more and more creative. This is Harris Wang, who points out that there are a lot of essential metabolites that some organisms can produce, but we mere mortals require these vitamins by intaking certain food sources. So we can't produce some of our own vitamins. So let's fix that problem. Let's chromosomally introduce biosynthetic pathways that produce all of the essential metabolites that we can normally make from simple sugars, like from pyruvate in central metabolism. So they'll test maybe a couple hundred kilobases of DNA and do various configurations. And then as a test bed, they'll use cell lines to define media. So yeah.
Why? I mean, it's not that expensive.
Yeah. The vitamin companies are probably just horrified by this. Like your daily gummy bite or whatever. So one potential would be cell culture media is a little bit expensive. So I guess this would avoid, you would have very defined media. So maybe you wouldn't have to supplement your media with anything. I think avoid FBS. That's an expensive reagent. It's also an interesting kind of, I guess you could also do this in plants, right? Like what if you wanted to take a plant that exists, it's currently a staple crop, and then have, actually no, never mind, I shot that it was down. Yeah, what do you think?
One thing people are now talking about, like lab grown meat. So rather than growing the organism, if you have the prototropic meat, you can just grow it in the lab, and like you said, no FBS needed. Makes it a lot cheaper to do that.
But then why humans?
Sure. Was there another question?
So I think I have a partial answer to Sarah's question, why human? This is funded by DARPA. And so I think the way they had to write this grant proposal was like, human soldiers need all these vitamins.
We have our answer. We're gonna have super warriors that are indestructible. No, sorry. This is exactly the kind of important conversation that should be had in the way that I'm not facilitating. So yeah.
I was just gonna, I mean, actually we were just having in the drill down this morning a conversation about engineering yeast to produce B12, because B12 is one actually that it would be nice if you could just make, so vegans have no way to get B12 at all. You have to take a supplement. So if you, but vegans do, for example, consume like nutritional yeast, but the one vitamin it doesn't have is B12. So if you can make yeast, make B12, you don't have to do that industrial process anymore, you know, and you can just make it in yeast extract. That would, I mean, I bet a company that makes yeast extract would actually pay a lot of money for that. But again, that, you know, this probably is just a cover for figuring out how to do yeast, right? Like, you know, Eric Swan probably doesn't really want to make human cells make B12. He wants to make yeast make B12 and then make a lot of money off it.
I do know from Twitter that I've learned rumors that the Harris Wang Lab and the prototypic human genome people are talking to like the cellular agriculture people.
Yeah.
So
yeah, this is just so that DARPA gives them the money to make yeast to it. Like that's why they're doing this.
So it's interesting because like this is a true statement, but like yeah, so everything's true, but altogether there's something missing. So maybe now we know. Maybe it'll be yeast. Thanks for that insight. Okay, this one's more of a fundamental science thing. The whole lab, they basically argue that during embryogenesis, a lot of the tissues and patterns that form in the final organism really only involve 7 core signals, just a handful of instructions that combinatorially and temporally encode a lot of information. So they want to essentially map the signaling space for different differentiation routes for a variety of human cell types from stem cells in vitro, using optogenetic and chemical control, all buzzwords in one sentence. So basically, they want to think about the human or the genome project right as an opportunity to explore how you control differentiation very precisely for eventual applications, maybe tissue regeneration or replacement. So I think this is something that's interesting as a spin-off that maybe wouldn't have come to mind immediately, but synthesizing a genome is to how to think about controlling differentiation. But I guess it falls under the same umbrella. All right, and then, oh yeah, back to crops. So it would be very useful if you were going to genetically engineer a crop and then introduce it into the wild to try to avoid it from recombining or making hybrids with other existing crops. Plants do this all the time. They combine their genome, very high cloning number. So if you characterize a synthetic organism really well, and you were very confident about how it behaved, and then it went out into the wild and made a hybrid of something else, that would be something new that you hadn't characterized before. So that could be a little bit scary. So in the Ryan lab, what they're trying to do is, using yeast as a model system, scramble the features of chromosomes, similar to what the Silver and Way were doing, but basically to prevent any plants from making hybrids. Another example, safety feature is you could have all these different combinations of pairwise toxin and antitoxins, wherein if a chromosome is ever separated from what else it was supposed to be with, every resulting cell from that division would just die, because it wouldn't be able to have the antitoxin for the corresponding toxin. Basically, just trying to make genetically engineered crops safer. Okay, so that's just, I guess, a brief overview of some things that have been approved. This next group are things that have been proposed. And I very much appreciate their openness in even putting this out here, because they're under no obligation to talk about proposed projects, right? Okay, so it's pretty easy to evaluate a gene if there's one copy, but since we're diploid, that's a little tricky. If you're using CRISPR to knock out a gene, you have to consider both copies of it. So there are certain types of cell lines where in culture they're haploid, and it's easier to study, but those haploid cells can very readily become diploid, just kind of spontaneously. Eventually the diploid cells overtake the haploid. So this group, the EGN group, wants to determine the mechanism by which this is happening and try to find a way to avoid diploidization. So this is, I guess, another more tool or fundamental science question about the mechanism by which chromosomes come in different ways. Oh, this is a bit tautological, but they add, as an added perk, you only have to synthesize half the DNA.
I feel like that applies to the plant genome projects as well.
Yeah.
They're nightmares, like huge and tons of copies.
Yeah, yeah. It's kind of like, we're going to synthesize half a genome, and as an added perk, we only have to synthesize half the genome. Okay, who gave a talk on...
By the way, do they have a project to remove all the junk DNA?
Can you say more now?
Do they have a project to remove the junk DNA from the genome?
The junk DNA. I don't know, like, so there's a minimal bacterial cell, right, from the Venture Institute, but is there like a minimal human cell being proposed?
That would also quite reduce the amount of DNA you need to synthesize.
It would. I don't know. I have not seen a project that proposes that. But something like haplogenomes, genomes, remnants of viral integrations over the past few billion years, right? So they might do something. I don't know. I haven't seen anyone propose to make minimal human genome.
So if ChurchLab is only making exons, maybe part of that will just be like assembling all the exons and seeing if it is happy in a cell.
Yeah, I don't know. The word junk DNA is a misnomer in itself, right? Because it was labeled junk DNA before ENCODE, before people started thinking about the more nuanced roles of non-coding regulatory agents.
Well, there is a big controversy. There is a nice paper from Denver.
What was the last word? I'm sorry, I didn't hear.
There is this controversy about the amount of genome which is functional. There is this paper from Denver.
Oh, okay. Yeah, I don't know. But I haven't seen anyone tackle that question yet, at least in this project space. On another topic, who was it who gave a talk about encoding information? Like Catherine?
Yeah.
Okay. Yeah. Well, this may be a follow-up on that. So, DNA storage, kind of treating DNA as a computer. This group wants to, computer science professor, wants to encode an entire computer operating system. Also, just for funsies, also moving some other files all into DNA. And be able to retrieve this info many times accurately using Muldoon sequencing. Right? So this is a two-way street. You have to encode into DNA, but if you can't retrieve it many times, then it's like you can only open the file 10 times before it deletes, right? Kind of equivalent. So they want to avoid this challenge. So in some recent preliminary work, they've been thinking about the limitations for how much info people can really store in DNA. What's the density of packing that information in? And so far, it looks like 200 times 10 to the 15 bytes per gram of DNA. And that's the highest you'll get achieved by over an order of magnitude compared to what anyone else has up till now reported. So that looks pretty promising. I suppose that must be more than a computer, right? On the order of, that's what they plan to do. Do people feel like this, like the public would be less perturbed by this kind of a project?
No. I mean, it's already happening, right?
Yeah, but if you present the option of encoding a computer in DNA versus encoding an organ in DNA, do you think people respond differently?
People would probably think like computers with beating hearts that are like under their desks.
Okay. I mean, you could do that. We could like have
But you should.
the computer chromosome be the 24th chromosome. Make it organoid.
So Joe, I was wondering, I know there was a huge backlash when this was initially proposed, in part because of how the meeting was held. Do you know how much of the rebranding from HGP right to GP right was sincere in terms of trying to get at multiple different kinds of genomes versus just a PR problem?
I wish I had the inside scoop. There are certainly a number of proposals, so I'm focusing a little more on the science, but there are a number of proposals that come from philosophers and bioethicists that have been both proposed and accepted for funding. So that's definitely an important component that at this point is being explicitly held up on the same level as every other type of project, I presume. So that bodes well. I don't know in the intervening 2 years what exactly took place together.
Address the question of using the public market, and it relates to the rebranding. Like anything not human, I feel like probably just selling. At least, like you guys like my parents, right? It would be like, we're not. Like I think that there was something that really stirred a lot of people about human. So to me, this is my list.
Do I agree?
Yeah.
Oh no, I'm just the presenter. Not gonna comment. Do I agree with what the public feels, or what do I feel?
Well, that was just.
Oh, yeah, no, I could imagine that if there were a trend, that sounds plausible.
Considering how non-receptive the public is to genetically modified organisms, I can imagine that they'll be too receptive to genetically modified
I think at the same time, like Twist and Microsoft and the people who are already working on this are like, I think in their PR trying to make it palatable. Like we put in an OK Go music video. Like look how like cool and hip we are. I feel like the PR that they're doing is making it a little more palatable to the public. Although as someone who knows more about this, I don't really know. I would have to ask a friend and show them the press release and be like, what do you think about this? But I'm not sure.
I feel like this would be like incredibly easier to like handle for the public because a lot of the issues I believe with like the whole organism, like your messing up life and all the other type of stuff. But this is just using DNA as a tool. So I don't think it wouldn't be that hard to stand.
It's kind of funny because 20 years we've been encoding people into computers, right? That's when you go to NCBI and you look up the human genome. So it's just the other way around. Conceptually, shouldn't be that problematic, maybe.
But also not GMO stuff, right? So
yeah. All right. So just one more thing. This is an example of a bioethics study that's looking to compare policies across different countries. And so they're basically just trying to really drill down and ask, for a lot of the policies that are in play in different countries, how will this impact the way that people can work towards the goals of GP right? What are kind of the legal regulatory measures that will be required in different nations or different cultures in order to kind of facilitate this uniformly? And then are all the principles that underlie these consistent? Are we consistent in we make a decision in one part of the world for what research ought to be allowed? How does that work? Oh yeah, and there's some popular press on this. So Wired magazine, as of I think a week or two ago, interviewed George Church, and they have a whole thing on this that you can read. And there are 9 working groups. You can see, I think, back to Sarah's point, they have a dedicated working group for communications and public outreach, as well as for education and for the ethical legal side of it. There's Booker Church, Hessel, and Kelly. So I guess that is when we might hear the updates about where this is all going. That's all I have.
I mean, how it's.
I felt like
Please remember to sign in, if you do not do that already.
It's a really interesting So Drew Indy's objections to the human stuff are like, number one, focusing on humans takes the attention of all these brilliant labs away from, like, more pressing issues like environmental culture.
Alright, so I'm going to present the newsreel today. So this is Opentron's new lab robot, so for lab automation. It'll set you back about 5K, but the advantage here is that this is the first one to use a very nice GUI instead of having to learn Python or some other coding language to do it, and they just had a $10 million funding round for more robots. So when you are running a lab, this will be your grad student. All right. Next, I want to talk about SynLogic. It's their company that does bacterial-based microbiome therapeutics for metabolic disorders. And so they did their first human dosing this week, or recently. I don't know when, but so the first human dose, and this one is for liver failure. And so the bacteria have this synthetic gene circuit that convert ammonia into arginine and excrete it. They have like 8 other drugs in the pipeline. Their website is really cool if you're interested in microbiome therapeutics. So the next one is Join, which is a joint venture between Bayer and Ginkgo to work on engineering bacteria or probiotics for plants. And so, for instance, they've got some nitrogen fixing ones that will help things that can't fix their own nitrogen, fix nitrogen. All right, this one is kind of cool. So this is Wild Earth, and this guy is going to make a fungus-based dog food. So his quote here is, Wild Earth will be the first to bring cultured protein and cultured meat products for dogs and cats to the market. They're nutritious, humane, and without the devastating ecological impact of factory farming. And using biotech will give us the ability to scale and to get a product to market safely, quickly, and affordably. So if you like paying a premium price for your dog food, this is where we go. So Modern Meadow, which is the synthetic leather company, has recently partnered with the fermentation company Evonik to scale up production of their biosynthetic leather. A couple papers this time. So this is raised from the Jewett lab on cell-free protein synthesis with non-standard amino acids. Also in the non-standard amino acid field, they were able to, a different group was able to incorporate non-standard amino acids in silkworms to keep the silkworms alive. That's pretty cool. The Baker lab is up to their usual stuff. They have designed more really cool proteins. These ones are multi-pass transmembrane domains, and there are a lot of them, a lot of really pretty figures in that paper. Let's see here, what is this one? So this is the efforts of a big genome mining project in bacteria, and they found all of these promoter elements and characterized them across a couple of different species. And so now we have a much larger library of programmable species-specific promoters. All right, here's a cool one. So Tim Liu's group making multiple biologics from a yeast culture. So one yeast that has a bunch of different chemically inducible circuits in it for production of antibodies and other biologics. Now, if you were to use them separately, you could do that, but this could just go ahead and produce the combined dosage in kind of one fermenter and one purification. The last one, this is evolved Cas9 variants, and so they've got a broader PAM compatibility, so more places in the genome are now accessible, and they also have higher DNA specificity. They use this process called PACE, which is phage-assisted something evolution.
Continuous.
Continuous, there, that would be it.
All right, and got some new cast iron elements, so that's pretty cool. And then the last one is, this is a bunny cat. Hang on. Yep. Bunny cat, so thanks for coming. And, just as a reminder, our monthly meetings are now the first Friday of the month, typically. That might be false next month. But we kind of shifted to avoid conflicts with the CSP seminars, which are towards the end of the month. All right, and then Adam's got a couple things to do.
I wanted to just put up a couple of announcements. So yeah, first, next month we are actually the second Friday of the month. So that's really fun. We actually negotiated, there's a local bio artist that's going to be coming and give a talk. He wants to do some, both interact with GeneMods and then also give some ideas of how he wants to use synthetic biology in his work. A couple of other things. Was it Katarina, you want to come up here? Or do you want to explain your thing? Or?
Just have a spreadsheet that I wanted to helping the action team brainstorm ideas.
It's really great. If you want to help out the iGEM team, they're doing a bunch of things. They want to get some ideas for what they're going to do, and I imagine later on in the summer, are you still looking for people for like training on the SMA?
It's kind of going to be a little different this year, so.
Okay, but if you want to get involved with GeneMods, talk to Katarina, talk to Vaughn. Sorry. Okay. Get involved with iGEM because it's awesome. Okay. Two other quick announcements. So first off, I just wanted to do a quick poll. We're getting toward the end of the year, and for those of you that are first years, especially, you've sort of seen how GeneMods operates, but we wanted to kind of get an idea of what y'all wanted from this club. Right now, we're kind of set at doing our monthly meetings where we have, one or two grad student talks or an external talk from a faculty or, like I said, an artist or an ethicist or something like that. I'm sort of opening it up before, though. We've had kind of internal discussions on whether or not we should do social events like a GeneMods happy hour. One thing that we sort of ban is people talking about their own research at these talks, which I think is great because it forces you to go outside your comfort zone. But we were talking maybe it'd be fun to have like either a lightning talk competition or like a 5-minute speaking competition where we have like a traveling lab trophy for who can do the best one of those. If anyone's got any particular ideas on that, I would love to open up the board to it. I'm not sure how much time we have.
You can also do like a Google form.
Yeah, so we can do a Google form and send that out. I'd love your ideas. Even if it's just something as easy as like a happy hour for all the synthetic. I mean, we already do that, right, in the department, the CSB labs. I know there are people here that aren't in the Center for Synthetic Biology, and I mean, a lot of us are friends. It might be a good way to sort of interact and also just learn about people here might know something about the technique that could help you in your own research. We've also talked about doing a site visit to some local biotech area companies. That obviously is going to be contingent on whether or not people want to go. So hit us up if you're interested in doing that, and that'd be great. Also, if there's any particular external speakers that you're interested in hearing from. So mostly we do academics, but if there's industry people that you'd like to hear from, or specific subjects, oh, I'd really like to hear someone come in and talk about like sequencing, then we can do our best to try to come up with that. Interfacing well with like both the department seminars, the BTB seminars and everything. There's a lot of different options to hear about synthetic biology on this campus. The last thing I wanted to bring up is that we are inviting people that are interested in joining the GeneMods executive board. Our kind of aim is to start turning that over, starting over in the summers, that so we can hit the ground running in the fall. So if that's something you're interested in, the current board is me, Gordon, and Patrick. But we are always happy to have more, if only because you present the newsreel with 1/n frequency for the number of people on the board. So that dilutes that responsibility. But other than that, pretty thin. So if we wanted to invoke some of these more social events or external people to come in, it'd be really nice to have enthusiastic new members. And that's especially a plea for the first year grad students that want to get involved. Undergrads, like
Jordan is an undergrad, guys, if you didn't know that. She's like the most knowledgeable person about synthetic biology. So please join. It's a lot of fun. It also gives you the opportunity if you want, and you don't need to be on the board to do this either, to participate on the podcast. That's a lot of fun if you don't listen to those. And by the way, announcement for that, I think there's an interview with David Baker that Isaac and Jordan did that should be out.
It'll be out probably in the
next couple days. So that's really exciting as well. But yeah, again, we'll probably plan on starting that around the summer. If you're not going to be here this summer, that's fine. We'll work with it. But hit us up if you're interested in doing that. And I think that's all I had. Please sign in so we can get the prompts up. And I'll also the iGEM sheet as well if you're interested in helping with that. Okay. I'll make a. I know, right? This moving.